It's a pleasure to be here. I've been giving this lecture for about 8 to 10 years now, and it's really one of my most favorite meetings and groups to lecture to. It's wonderful to see you all. And as I mentioned always in my lecture, that Claire Ann just does such an outstanding job of telling you about biological memory that it makes my work really easy. And so what I'm going to do today actually is touch upon two subjects. And both of these have been mentioned actually before in the previous lectures. And one is about T follicular helper cells. And these are the cells which are critical for generating optimal antibody responses. So in the first part, I'll describe some features of these cells, present to you some data from our lab looking at the role of these CD4 T helper cells, and then address a question that's of interest in terms of not only basic biology of CD4 T cells, but also in terms of vaccination. And that question is there a memory cell that's committed to become a TFH cell? Is there lineage commitment of CD4 T cells? So we'll address that issue. And then in the second part, I'll switch to CD8 memory T cell differentiation. And here I want to tell you about a cell which again has been mentioned, but probably not much spoken about, which are tissue-resident memory cells. These are really your first line of defense against invading pathogens. In the lecture on mucosal immunity by Adam, there was a wonderful discussion about mucosal immunity and IgA, but there's also a specific type of CD8 T cell that doesn't recirculate in the blood, but is sitting, and these cells are referred to as tissue-resident memory cells. So I'll describe some work on that. This work actually is not our work, but I take pride that this is one of my trainees' work, Dave Massapust, who's at University of Minnesota, and I borrowed a few slides from Dave to share with you some concepts about tissue-resident memory cells. And then I'll end the CD8 memory T cell part on some work that we've been doing over the past 5 to 10 years or so, looking at CD8 T cell responses after yellow fever vaccination. And again, you've heard about yellow fever vaccination, and the real correlate of protection against yellow fever actually is neutralizing antibody. But this is also a wonderful model to study primary CD8 T cell responses to an acute viral infection in humans. So I'll share some of that data with you. Okay? So basically 3 parts. The first is CD4 T follicular helper cell part, then in CD8 T cells about tissue-resident memory cells, mostly mouse studies done by Dave Massapust, and then our work on human CD8 T cell responses to yellow fever vaccination. Okay. I think Adam or someone showed a very similar slide. But what this shows you is that, is that CD4 T cells come in many different flavors. Unlike CD8 T cells, which are actually kind of a single lineage, they're killer cells. They're always killer cells. They become memory cells, they don't kill, but they always have that one single program of killing. CD4 T cells are very diverse in their lineage commitments. So you can have these T follicular helper cells. You can have Th1 cells, which you've all heard a lot about, or Th2 cells, Th17 cells, and Treg cells. And what kind of is critical in giving you each of these lineages are transcription factors, key transcription factors. For example, BCL6 is a key transcription factor that encodes genes that TFH cells do. TBET is for Th1, GATA3 for Th2, ROR gamma T for Th17, and of course FOXP3 for Tregs. Okay? And if you get a bacterial infection, for example, very often you will get a Th2 type response. Several fungal type of infections will give you Th17, and of course Tregs are regulatory cells. Almost all viral infections will give you a Th1 type response. But the TFH response is induced after every type of infection, because you need antibody for every infection. So viral infections will give you Th1 and TFH. Bacterial often will give you Th2 and TFH. You can get Th17 and TFH, because almost every pathogen, you need to have antibody against it. So we've evolved a mechanism where, whether it's a bacterium, a virus infection, or a fungal infection, you will always make some antibodies. So the TFH response is something that's consistently seen after every microbial infection. So what are these cells? So the main function of these cells is they're essential for all T-dependent CD4 responses. They're essential for help in germinal center formation and maintenance. And I guess you all know what a germinal center formation, what a germinal center is. What is the main function of a germinal center? What is the most important thing that happens within a germinal center? There are two important things. What's the first one? CD4 and B cells come together. There's this anatomical feature in the lymph nodes where the vaccine of the infection is, okay, or in the spleen. What is the major function of, major thing that happens? Affinity maturation, exactly. So in order to get high affinity antibody, the germinal center reaction is absolutely essential. Okay? That is the single most important reason for that. And then there's a second very important reason for germinal center formation. All of you may not know this, but some of you might. Memory B cells come out of germinal centers, that's correct, and partly related to also having higher affinity maturation. But there's one other thing. There's one other cell that requires, one other critical cell that requires the germinal center reaction. One, of course, affinity maturation, memory B cells coming out of it, that's the other one. There's a third critical cell that the GC reaction is critical for. How do you maintain antibody for many, many years?
Follicular dendritic cells?
No. How do you sustain antibody production 10 years or 20 years after your vaccination? That's right. So the germinal center reaction is essential for generating the long-lived plasma cells. Okay? And for all of these things, the CD4 T cells are needed. No CD4 T cells, no germinal centers, no affinity maturation, no memory B cells, no long-lived plasma cells. Okay? And so how do you define these cells in terms of phenotype? If you're studying them, the canonical marker is CXCR5. Do you know what is CXCR5? It's a chemokine receptor, right? And it sees a chemokine that's produced in the B cell regions. Okay? It's the chemokine is known as CXL13, but that's not important. It's the chemokine that is produced where B cells are is, and in the follicular regions, only cells that express CXCR5 will go there. So this is a Th1 cell does not express CXCR5, a Th2 cell does not express CXCR5, neither does a Th17 cell. It's the TFH cells which will go into the follicle, go to the border of the T and B cell zones, and then go into the border that will express CXCR5. Okay. Which other cell might express CXCR5? What's the other cell that's in the, what's the other important cell that's in the germinal center? B cells. So both B cells and TFH cells express CXCR5. Okay. So it's a common marker on, in fact, CXCR5 expresses is even higher levels in B cells. Okay. And as I mentioned to you, BCL6 is a lineage marker for differentiation of TFH. Okay. Other markers which are often present on CXCR cells is ICOS, PD1. And in terms of cytokines, they produce several cytokines, they make some other things. So but the key cytokines they make are IL-4 and in particular IL-21. Okay. So the question that we asked, and I'm shifting here now a little bit to some work that we did in our lab several years back. Okay. And the question we asked is that at what stage of a B cell response are CD4 T cells critical? Okay. So we said, are they needed? This is just a cartoon slide showing you what would happen after an acute viral infection, but this cartoon would hold true for any acute infection where you look getting a T-dependent response. So we asked the question, what would happen if, and I'll walk you through this in a second, but there are three different phases where we depleted CD4 T cells. We asked what would happen if we deplete CD4 T cells in the beginning, in which case, of course, you abort the whole T and B cell response that is T-dependent. And then what would happen if you deplete CD4 T cells at a much later stage, and what if you deplete them in the middle. So here's just a cartoon thing showing you the kinetics of how different responses happen as far as the humoral response is concerned. So in an acute virus, I've removed from here the CD8 T cells just for simplicity. Of course, the CD8 T cell response would also be made. So here's the virus is cleared within a week. Okay. There is a very vigorous antibody secreting cell response that you detect very quickly in the spleen. And this is actually extrafollicular plasma cells which are formed typically. In the first burst of plasma cells you get after a primary infection or primary vaccination are mostly extrafollicular. Now, this is still T dependent, but it's not inside the germinal center. This actually is before germinal centers have formed. And it would be in the draining node or in the spleen. Okay? And the first burst of antibody that you get is not due to B cells, plasma cells that come to the germinal center, but really first burst comes from the splenic or lymph node, the extrafollicular ASCs that you get. Germinal centers start forming typically around a week or so. They peak around 2 weeks post-infection, and then gradually decline in the next few weeks. So you have germinal centers for about 3 to 4 weeks. Again, memory B cells will come out of the germinal center, so you have them coming up later over here. And the fact that memory B cells come up later is one of the reasons which I think Claire Ann mentioned, is that when you're trying to boost, you want an interval between the first and the second. If you try to boost too early, basically you really don't have a good pool of memory B cells. So that's why there's an interval between the prime and the boost for antibody, because what you want to boost is really your antibody, is your memory B cells. And then the splenic, or this initial burst of response to the spleen, antibody secreting cells in the spleen or the lymph nodes come down. Okay? And then from the germinal centers, you start getting the long-lived plasma cells, which actually migrate to the bone marrow, and then these can be retained for extended periods. So the question we asked was, what would happen if CD4 T cells here? Of course, you abort everything. You don't get a good antibody response. Okay? All of these things get aborted. What do you think happens if you deplete CD4 T cells at this stage, where everything is now homeostasis, that is, the virus has been cleared, germinal centers have disappeared, there are now long-lived plasma cells in the bone marrow, which are secreting antibody. Okay? You have memory B cells in circulation. What would happen with CD4 T cells here? Will you lose plasma cells, or will you lose memory B cells? Would you lose serum antibody? No, you don't, because it's on autopilot right now. Okay? But what would happen if you deplete CD4 T cells and then did a boost? You're not going to get a good response. Okay? So again, when there is a re-challenge or reinfection, okay, you again need the CD4 help to recapitulate this whole this whole pattern of T and B cell responses. Okay? What do you think would happen if you depleted CD4 T cells during this phase, while germinal centers had already formed, and if you then deplete CD4 T cells, what do you think would happen? Huh? Yes, and actually it's quite striking. What happens is that if you deplete CD4 T cells, let's say at this peak of germ formation, almost immediately the germinal centers just dissolve. Okay? Within 24 hours to 48 hours of depleting CD4 T cells, germinal centers just decrease. There's a big drop in the germinal centers. Here it shows that there was high number of germinal centers at like day 14 after infection. When you depleted CD4 T cells, immediately the germinal centers went down in the mice with CD4 T cells were depleted, where they stayed up as would be the normal course of the event. So not only are CD4 T cells necessarily initiating it, but then to sustain the GC reaction, you need CD4 T cells. This is just a histological photograph showing you what I referred to as the ruins of germinal centers after CD4 depletion. So this is looking at the germinal center area. They're just showing the B cells over here. But the bottom line is that here was a beautiful germinal center, but then 2 days after treatment, basically all the germinal center reaction is pretty much gone. Even though we're not depleting B cells, we're doing CD4 T cell, that whole formation, and then has consequences on the amount of long-lived plasma cells you get, definitely maturation you have, number of memory B cells you get. So what I've illustrated from these very simple slides and experiments is that CD4 T cells are absolutely critical in giving you all of the essential things that you need for serological memory. Long-lived plasma cells, high affinity maturation, and for having extended, having a large number of memory B cells. Okay? And also this needs to be a sustained reaction. That is, the TFH have to be active for at least several weeks in order for this germinal center reaction to continue. Okay? So what is important in germinal center reaction formation? okay? So one of the important things is IL-21. So if the TFH cells don't produce good IL-21, and work has been done by us and by many other groups who have shown two defects. If IL-21 is not there, either IL-21, the substance have been done using either IL-21 receptor knockout mice or IL-21 knockout mice, and the end result is the same. You get suboptimal germinal center formation, okay? But more strikingly, you do not get the long-lived plasma cells. In some situations, germinal centers do form. They're not as robust and they're not as nicely maintained as when IL-21 is there. But in every model that has been looked at, whether it's an infection model or a vaccination model using a model protein antigens, okay, you do not get long-lived plasma cells if IL-21 is not produced by the TFH cells. And this is just an example of some of our experiments. So this is infecting mice with the acute infection, in this case with LCMV, but we see same results with influenza or with VSV. Looking at antibody in the serum, the wild-type mice generate a response and maintain for extended periods. Okay? This is going out about a year after infection. Remember, the virus is cleared within a week over here. And this is showing you the IL-21 receptor knockout mice. So you get this initial response of antibody. So where is that coming from? Where is the initial antibody response coming from? IgG? Extrafollicular, right? So for the extrafollicular T-dependent response, plasma cell response, IL-21 is dispensable. Okay? But to give you the germinal centers, the high affinity B cells, and the long-lived plasma cells, IL-21 signaling is absolutely essential. This is just an example showing you that this is now looking at virus. Just look at the bottom one. This is looking at virus-specific plasma cells in the bone marrow, and you can see that basically the white bars are the IL-21 receptor knockout mice, the black are the wild type. Essentially, you get no plasma cells in the bone marrow. This is the spleen showing you that initially you did generate that short-lived plasma cell response in the spleen, but really nothing is in the bone marrow. And that's why you got this, this rapid disappearance of serum antibody. So basically, the short-lived plasma cells are generated, but the long-lived plasma cells absolutely need IL-21 signaling. Okay, now I want to address the question of, is there a TFH memory cell? So what I've shown you is that following infection or vaccination, you will generate TFH cells, which have the canonical transcription factor BCL6. They secrete IL-21. They provide help for B cells for doing all the wonderful things that are needed to give long-term protective immunity. Okay? And then what happens afterwards? So these canonical phenotypic markers which are used, such as high levels of PD-1, high levels of ICOS, high levels of CXCR5, high levels of BCL6, which we use to identify TFH cells, really are cells which are in the follicle, in the germinal center. So what happens to them after the germinal center disappears? Do you have TFH cells as memory cells? And if so, what markers can be used? Okay? So do you have memory, and more importantly, do you have a memory cell now which is committed to become a TFH cell? Okay? And why is that important? Why is the commitment part important in terms of vaccination strategies? Yeah. So let's take a hypothetical example. Let's say that if you had a situation where, I'll come back to the end. That point will be made better when I come to the end of this. Okay? So we ask the question, is there lineage commitment of these cells? Okay? And again, we use a mouse model of acute infection to look at this. And I'll give you some of the answers without the data. So what are the markers that define TFH memory cells? Actually, some of the markers are no longer any good. So if you were to and where are they found? So I'll tell you where they're found. So TFH memory cells in the mouse are found in the blood, and they're found in the lymphoid tissues. They'll be in the lymph nodes, and they'll be in the spleen. They will not be in any of the non-lymphoid tissue. So for example, if you look at a Th1 cell, you will have Th1 cells not only in the blood and in the spleen and the lymph nodes, but also some in the liver, some in the lung, and so on, okay, in both lymphoid and non-lymphoid. The TFH cell does not go to non-lymphoid tissues. It predominantly circulates within the blood, lymph nodes, spleen, and traffics through the bone marrow. It's not a major reservoir, traffics through the bone marrow. Most of them would be in the blood, spleen, and the lymph node. And how do you identify this cell? It still expresses CXCR5, so that continues to be a good marker. PD1 is no longer a good marker for it because PD1 comes up upon TCR stimulation. So if you're looking in the blood, you're looking many months or many years after vaccination or infection, PD1 is not the best marker to look for a TFH cell, but CXCR5 is. BCL6 also is not a very good marker because BCL6 levels go down quite substantially. They come up upon, again, B cell receptor stimulation and will be very high in germinal center TFH. But once they've come out of the germinal center, those markers are at a much lower expression level. ICOS is also much lower. Okay. But CXCR5 continues to be a reliable marker, although its level goes down. So you can use, and work has been done both in mouse and in human, showing that CXCR5 positive cells are in circulation, which have memory properties of TFH cells. So we then ask the question, so if you have a CXCR5 positive and a CXCR5 negative antigen-specific memory cell, would it then, upon recall, be committed to giving only TFH and only TH1, or would it be pluripotent? So that's the question that we asked. So don't worry too much about all the markers, but we use two markers, again, CXCR5 and Ly6C, but just focus on the CXCR5, CXCR5. So what is done in this experiment is we isolate antigens. Everything I'm showing you here is gated on antigen-specific CD4 T cells for the virus that we are studying. Okay? We know the epitope, we can use tetramers and identify these cells, or we can use transgenic cells. You get the same result either with transgenic cells or using MHC class II tetramers, using the appropriate peptide. Okay? So we isolate from a mouse that was immunized several months back. So this is now at a memory stage. You isolate the CXCR5 negative cells, these are TH1 cells, and then we actually ended up having two subsets of CXCR5 positive cells, but don't worry about this. Basically, we are looking at TH1 cells, which are CXCR5 negative, and TFH cells, which are CXCR5 positive. So these cells were then adoptively transferred into a naive mouse, and then this mouse is re-challenged with the same infection. And then we ask the question, do these cells differentiate into TFH? Do the CXCR5 positive cells give you predominantly TFH and the CXCR5 negative give you predominantly TH1, or do you get pluripotency? Remember, the naive cell is pluripotent because from a naive cell you got both. The naive cell gave you TFH and TH1. Now you've got TFH memory and TH1 memory. Now what happens? If you hit a TFH memory with antigen again, will it only give TFH cells and the TH1 only TH1 cells, or will it be pluripotent again? If it's pluripotent again, then probably not much to worry about, okay? Because always you'll get everything that you want. But if it's committed to one lineage or the other, then there are very important implications for vaccination. And the answer is that there is commitment, okay? And I'll show you the data there. There is commitment, and in particular, the commitment is very striking for TH1 cells. Once a cell has gone towards the TH1 lineage, which is the T-bet, driven initially by IL-12 cytokines and so on, innate signals, T-bet expression becoming TH1, okay, that cell does not, upon being hit with antigen again, give you TFH cells. Okay? And if you have TFH memory cells, they are strikingly biased towards giving you TFH memory again, but there is a small level of pluripotency in those. And I'll quickly walk you through the data. Okay? So let's look at this thing here. We're asking how many, after you transfer the pure memory cell population, either TFH memory or TH1 memory, hit them again with the virus, what % of cells become TFH cells? Okay? So if the cells were CXCR5 positive, these two, almost 60 to 80% of the cells now are committed to the TFH lineage. But if they were CXCR5 negative, that is, and they were the TH1 lineage, very few, only 10 to 20% give you TFH. Okay? Majority of them will become TH1. And the reason for this is because these cells now express very high levels of T-bet. These will express and very low levels of BCL6, and the converse is true if you have the CXCR5 positive cells. Okay? And this is, and the reason for this commitment, I'm not going to show you all the data, but the reason for this commitment is twofold. One is the transcription factors that they're now poised to re-express. And the reason they're poised to re-express it is partly because of other cofactors that might be there, but also because of major epigenetic changes in these cells. The point about epigenetic changes was mentioned earlier by one of the speakers. Okay. But when the cells differentiate and become a TFH effector cell, there are major epigenetic changes which are needed for expressing genes that are required for TFH function or TH1 function. And even after the protein has gone away, because the TCR stimulation has gone away, the genetic changes that happen, the epigenetic changes that happen, do not become fixed, especially genes open up by becoming either demethylated or closed down by becoming methylated. So these epigenetic changes are there. And this is really what imprints them going to a certain lineage and becoming either TFH or TH1 cells. Okay. I'm going to skip this slide. Okay. And so this is the model. Okay. So you have a naive cell, which is pluripotent, and it can either go, so in a viral infection, you're going to get both. You get TH1 effector cells, and you get TFH type cells. Okay. And then when you go into the memory stage, you have highly committed TH1 memory cells, which upon being hit a second time, really now give you only TH1. This pluripotency part has been lost. Okay. The TFH cells also give you predominantly TFH effectors again, although about 10 to 20% of these will go in this lineage, but they're not as effective as TH1 cells, have lower levels of granzyme B and so on. Okay. So why is this important from a vaccination point of view? Why is this commitment issue important? Yeah, so let's say the reason it's important is because now there are many strategies where people use different prime boosts. They use different vectors. For example, in the HIV field, in the TB field, many different vectors are being used. So let's say if a certain vector that you used was very biased towards just giving you TH1 and did not give good TFH memory cells, then the boost there would not be very good. So what you want is that if your goal is really to get good antibody responses, you have to make sure that not only is your antigen activating the right B cells, but also there's inducing either the right adjuvant is used or the right vector is used so that you get good TFH memory and that you don't overcommit your CD4 T cells to the wrong lineage. Okay? And this is a very fundamental aspect of T cell differentiation, which has profound implications in terms of optimizing and developing vaccines as we move forward into more difficult challenges. Okay, let me shift here and talk to you about memory CD8 T cell differentiation. This is just a cartoon showing you the kinetics of the CD8 T cell response. You have this big expansion when naive cells replicate very efficiently, very fast, become effector cells. And then you have this contraction mode where about 95% of the effector cells will undergo apoptosis, again, very nicely discussed by Claire Ann in the earlier part of the lectures today. And then you have this phase of long-term memory of the CD8 T cells. Okay. So what I want to address today is the type of memory cells that are there, the type of CD8 memory cells that are there. So what has been mentioned by Claire Ann today earlier is you've got your effector memory cells, which are a little bit more poised to becoming effector cells. These cells are found in both lymphoid and non-lymphoid tissues. Then you've got your central memory cells, which have very optimal proliferative potential, and they are mostly in the lymphoid system, not so much non-lymphoid. And this is fairly well described, this kind of pioneering work that Antonio Lanzavecchia and Federica Sallusto did many years back, and this is kind of textbook, this is textbook material now. Okay? But there is a third cell which has been more recently described, work done by several people, not our lab, but several other groups have shown this, and this is the tissue-resident memory cell, okay, which is different from your effector memory cell. The key difference is an effector memory cell recirculates back into the blood. The TRM are tissue-resident memory. They don't recirculate. And this is kind of a cartoon of typically when these TRM are generated. So You have a naive cell, gets activated, becomes an effector cell, and then is going to go on to give you the pool of memory cells, which will, and this memory cell compartment will contain both your effector memory and your central memory cells over here. But then what happens is that the effectors, if you look at effectors, CD8 effectors generated following an infection early on, they express multiple homing receptors. This was a bit of a surprise that we found several years ago. For example, if you look at day 5 or day 6, CD8 T cell effectors after viral infection in mice, they will express, even though they are in the spleen, even though the infection was not from the gut or anything, they express, for example, alpha-4 beta-7 and CCR9, which are markers that will take them to the gut. And a small fraction of these cells actually do end up in the intestinal mucosa. Okay? And then they further differentiate there and then will not recirculate. The pioneering work in this really was done by Leo Lefrancois in his lab, showing that these cells, that systemic infection, systemic vaccines will give you cells that will migrate to the gut because they express the appropriate homing receptors and then will stay there and will not recirculate. And what's even more interesting is that they then further undergo differentiation there to acquire a very unique phenotype. And that's shown in a cartoon over here. Okay. Sorry, so I'm ahead of myself. So not only, I just showed the diagram with the intestinal mucosa, but same thing is, so these cells have been observed in the gut, in the skin, the lung, salivary gland, so on. Okay. So basically, almost all of these external tissues you can find. They don't recirculate the other compartments, and again, not found. So when we are studying humans and looking at blood, this compartment is totally missed. We are not sampling. At no time in the blood will we be looking at the TRM cell. Okay? So what happens with these cells, this is work actually from Dave Massapus' lab. The earlier work was some of our work that Dave had done when he was in my lab, but this is all coming from Dave's lab. Okay? And what he's shown is that in this case, he again was studying intestinal mucosa. He's also looked at the genital mucosa. Similar things happen. Okay? That these cells, in fact, acquire a very independent program that's dependent upon signaling from several different cytokines, and in fact, like TGF-beta is involved in this, IL-33 is involved, TNF-alpha, and these cells acquire a very unique phenotype, and they look very effector-like. And so when it was found that, okay, these look very effector-like in the gut, for example, or in the genital tract, it was assumed that this was by the antigen being there, but it's not. It's kind of a little bit counterintuitive and hard to accept that how that can happen, but it's the milieu of the site, plus the cytokines which are being made there that really drive them to look very effector-like. That is, these cells will be full of perforin, they'll have granzyme, okay? But this is a cytokine-mediated differentiation at the site, which is still the mechanism of this is not worked out, but it's a very interesting phenotype and an interesting biology here, where much more needs to be looked at in terms of the actual mechanism. Okay? And these cells then sit there at the mucosal sites. They also have very limited proliferative potential. Okay? But they're very effector-like cells sitting, basically, they're your first, our real first line of defense, of course, is mucosal antibody. But then our second real line of defense at the mucosal surfaces are these tissue-resident CD8 T cells, okay, which upon infection of the first few cells will provide the first line of defense and of killing those cells. Okay. But I was always skeptical about this. I said, well, really, can a few cells just sitting out there without even any capacity to undergo proliferation, I mean, how useful are these cells? So when I would hear these talks, I appreciated the wonderful biology, but I was always skeptical about the real biological role in protection, because to me, numbers are important. Okay? Numbers are very critical. We talk about quality, but quantity is actually equally important in terms of protective immunity. So I was very skeptical that these cells really do something in terms of the first way of protection. But then a very fascinating paper came out by Dave Massapus in Nature Immunology last year. And what Dave showed in this paper is that not only do these tissue-resident memory cells kill the first few cells that get infected, but they also provide a wonderful alarm. Okay? So what they do is they will immediately, upon encountering, this is the resident memory cell here, it encounters, and this was studies he did looking at the genital mucosa. Okay? And so not only do they kind of can kill the first cell that is infected, but they secrete interferon gamma plus some other cytokines, which then induce a cascade of chemokines, and he hasn't defined yet which are the cells making all these chemokines there. But these chemokines then very quickly attract your circulating memory CD8 T cells into the site. So not only do you get the initial killing of the infected cells, but you get recruitment almost immediately, and this happens within a few hours. Okay? So within a day, within 12 hours to 24 hours, there's this influx of a large number of cells from the blood, which is where our memory T cells are. The central memory T cells and the effector memory T cells, which are in circulation, almost immediately are recruited into the site. This initial recruitment is not specific for the cognate antigen, because they're just coming by being attracted by different chemokines. But then the proliferation of the cells, once they get there, is highly antigen specific, because only the invading pathogen is there to activate those cells. So recruitment is non-specific, but then there is this massive expansion of the central memory cells, mostly, that then control this infection. So this now really makes a lot of sense to me. So not only is it providing the first line of defense for killing, but also serving as an alarm to recruit additional, the large pool of memory cells that we have in the blood, and that pool has those cells have terrific proliferative potential. Okay? So then they expand and then really provide, have the control of the infection at a very early stage. So this just shows cells coming in and controlling infection. So basically, if a simple way of looking at protective immunity by memory T cells is a wonderful division of labor. Okay? So you have the resident memory T cells, which are at mucosal sites. They will encounter the first infected cell that happens. Of course, they don't see the virus coming in because you have to only see the infected cell. So the very first infected cell that comes in not only results in that cell potentially being killed, okay, but also these cells will secrete the appropriate cytokines, induce this chemokine cascade that will recruit certain memory CD8 T cells in blood and lymphoid tissue. And then these cells, which come in within a few hours of this happening, can further expand because they have huge proliferative potential, okay, and differentiate into effector cells. So this is really the basis of protective immunity by CD8 T cells. Okay? And of course, when antibody is there, everything happens much more efficiently. Okay? All right, so let me now switch in the last 10 minutes or so and walk you through some of the studies we've done with yellow fever vaccination. Okay? So one of the best vaccines that we have currently, and which was made many years back, is the live yellow fever virus 17D vaccine. This is live attenuated vaccine. And this vaccine was made almost 50 years back or longer by Max Tyler. And actually, the first Nobel Prize given to a viral vaccine was given in 1951 to Max Tyler, who produced the yellow fever vaccine, which really has been of enormous benefit in terms of controlling yellow fever infection. So this is a wonderful vaccine which a single immunization can give you very long-lasting neutralizing antibody responses, and the correlate of protection is neutralizing antibody against yellow fever. But as I mentioned to you, for us, we use this as a system to study CD8 T cell differentiation in humans. Okay? So our lab, for many, many years, had done a lot of work in mice looking at CD8 T cell differentiation. We said, what about studying humans? And most of the human data for memory CD8 T cells comes from studies with chronic infections, HIV, human CMV, human EBV infections. So wonderful work done, but these are all really chronic infections. So our goal was to try and understand how memory CD8 T cells differentiate in humans to an acute viral infection, and we were interested in a primary infection, not in a recall response. Okay? And then also ask some fundamental questions about the longevity of this response, the persistence of this response, how do these cells look like in terms of the phenotypic properties, and so on. Okay? So again, ideal model for studying a primary, and why primary? Because You can give this vaccine to adults who have never seen yellow fever virus before. Okay, so we're looking at a primary response. Right? So by using this vaccine in healthy young adults, we have identified a lot of different CD8 and CD4 epitopes, but I'll share with you data of one particular epitope which allows us to actually track responses longitudinally in people for extended periods. So this is a response from one of the non-structural proteins of yellow fever virus. And this is presented by the HLA-A2 molecule, which about 30% of the US population has, actually 30% of all humans have. Okay? So basically everyone who's HLA-A2 positive, which is about 1/3 of you sitting in this room, will make a good response to this particular epitope. Okay? Of course, you'll make those of you not HLA positive will make a response to other ones. So the vaccine works in everyone. Okay? But here is a nice dominant epitope that we can track. And this is just showing that longitudinally you can track these out to a year or even longer. So this is just in the blood. You can see at the peak is about 2 to 3% of the CD8 T cell, this epitope, in some cases up to 5%, and you can track this out for long periods of time. So by tracking this longitudinally, one can ask many questions. One can look at the gene profiling of these cells you can do. You can look at phenotypic markers. You can look at their function and so on. Okay? And this is just showing you now looking at when these cells are proliferating very rapidly initially, then they come down. Activation markers early on, and then these markers come down. BCL2, which is usually downregulated effector cells, is then re-expressed in naive cells, CD127. But what I want to show you is one very important thing, which is that these cells, remember I told you about effector cells expressing homing markers, okay? So we looked at homing marker expression on cells which are in the blood early on, yellow fever specific cells. And what we found is that yellow fever virus specific cells, early effectors, which you find at day 11, express alpha-4 beta-7 and also express CLA, which is skin homing receptor. So these are early yellow fever infection. We're not giving the infection to the skin. Of course, this is not a gut infection. This is a parenteral vaccine. It's inducing effector CD8 T cells in the blood. And these cells express alpha-4 beta-7, okay, which potentially could take them to the gut. Very similar to data I showed you, like in a mouse model of an acute infection. Okay. Of course, we didn't go into the gut and look for these cells in these individuals, but the potential is there for these cells to migrate there. Also CLA, they potentially could go to the skin too. So effector cells early on do express multiple homing markers, not only in mice, but also in humans, with the potential to seed many different sites. So it's very likely that some TRM cells are also being generated by this yellow fever vaccine, some of which are going to the gut. Even though there's not a gut infection, they're still going there, and it's also, some of them are also going to the skin. Okay. I'll skip that. Okay. And then I want to spend a few minutes on two other things. One is about marker expression of certain markers which are used to put memory cells into different groups. And then I'll end with 2 slides about in vivo homeostasis of human memory CD8 T cells. Okay. So typically, there are 2 markers which are used routinely to divide memory cells into different subsets. And that's CCR7 and CD45RA. So naive cells, of course, are CCR7 positive, okay, and CD45RA positive. Okay. And usually your effector cells are CCR7 negative and CD45RO or RA negative. This is what you see. So the red here are your yellow fever virus specific cells at day 14. As expected, they are all CCR7 negative and 45RA negative. This is to be expected. But here is a surprise. Okay? What we find is that if you look later on, if you look at day 90, for example, and this is following in the same individual, that these cells now are re-expressing CD45RA. And we were very surprised by this finding, because what one would have expected is that you would be getting your effector memory cell type cells. What's the phenotype of those? This is textbook stuff. CD45, there'd be CCR7 low, and there'd be CD45RO, that is not RA. Okay? But we were very surprised by this finding, that the yellow fever virus specific effector cells, sorry, yellow fever virus specific memory cells actually were RA positive and still CCR7 negative. Okay. And this was putting them actually in a very different box. So this was putting them neither in the box of the central memory cells, nor in the box of the effector memory cells, but they were putting them in this box which has been reserved for what people refer to as terminal effectors. Okay. This is the box that textbooks use, vaccinologists use, everyone uses for defining cells. And we said, why in the world, when you are having this yellow fever vaccine, which is an acute infection, and is giving you this very nice level of persistence of these cells in the blood, why are these becoming terminal effectors? And this term, these cells which are referred to as TEMRA are supposed to not be very functional. Okay? And that's just data that comes from studying different kind of chronic infections and so on. Okay? And this is where all of them are falling. And then we ask, are these functional? Of course they're very functional. They will make any cytokine that you want them to make. They will make huge amounts of interferon gamma and TNF alpha. They make IL-2, they make MIC1 beta. They re-express CD107, okay, and so on. Okay. And so they're in the wrong box, and they also have great proliferative potential. Okay. And while we had published this particular phenotype of these cells, there was a paper that came out from Rick Kaub's group where he looked at vaccinia-induced, that is smallpox vaccinia virus-induced CD8 memory cells, and they also had the same phenotype. That is, they were CD45RA. Since then, about 4 or 5 papers have been published. One with our paper with the yellow fever vaccine, the vaccinia virus data from Rick Kaub's group, and then a paper with parvovirus, which is another acute infection in humans, and also with an acute hantavirus infection in humans. Okay. So this may seem a little bit heretical, but I think we need to kind of rewrite our rules here in terms of the phenotype. I think most acute infections are going to give you cells, most acute primary infections are going to give you cells which phenotypically are falling in this wrong box, but actually are highly functional cells, okay, and are not falling typically in the box that we reserve for cells that are induced by viral infections. Okay. And I was just talking to, when we had this result, I called up Julie McElrath, who's done many HIV vaccine studies. And I said, have you done your phenotyping to put them in different groups? And she said that actually for adenovirus, where they've done this quite nicely, most of the adenovirus-induced cells in human are also RA positive, not RO. Okay? So they're falling in that. So I think that we need to kind of there's a tendency to kind of box things a little bit too much by using a few markers, and we start boxing cells in there. And the conclusion would have been, well, this is a bad vaccine because giving you these terminal effects. The answer is no. If you do all the right assays, they're highly functional, but it looks like most acute infections actually will give you these RA positive cells. Okay, I see that Daniel is waking up. Okay. Standing up. So I will skip these. So what I've shown you here is this. So cells appear to pass through an effector that they transiently express homing molecules, not only to the skin, but also alpha beta 7, even though the vaccine is given subcutaneously. A window of opportunity for effector cells to migrate to mucosal sites after an effective vaccine. Okay. And one would assume that these will become then tissue-resident memory cells. Effectors are characterized by the distinct from that of the typical memory subsets. Okay. And again, I made this point to you that about not boxing them too much, and the importance of really not to give over-importance to a couple of markers, but actually to look at it in more detail. Okay. And I want to end in the last 5 minutes by asking one question. Okay. And we're actually quite excited by these studies. So this just shows that you can follow an individual over a long-term period, and these memory cells are maintained in the blood. Okay. And the question we asked is that how are these memory cells being maintained in the blood for over a year without much of a decline? There's some decline, but not much of a decline. And are we dealing with truly long-lived quiescent memory cells? Okay? That is, with a very long intermitotic period, or is there rapid replenishment and death going on every few weeks? Okay? And if you, by doing snapshot analysis of Ki67 or proliferation, you really cannot make that distinction. Okay? Because if there's a small fraction that's dividing somewhere, you really will not catch that. Okay? So we then designed an experiment to ask this question. Okay? And this is a study we've done with Mark Hellerstein, who's at Berkeley, who came up with this very nice in vivo labeling method in humans. So what is done is that the vaccinees actually will drink heavy water. Okay? So this is a labeling technique that Mark Hellerstein developed. Okay? So if a person is drinking heavy water, okay, which is perfectly safe, okay, with the deuterium which will get incorporated into the replicating cell DNA, and then by looking at the diaway of those of the deuterium, you can calculate the half-life of the cell. Okay? And you can do the deuterium thing by gas chromatography, mass spectrometry. Okay? So basically, you can ask turnover rates in humans by this analysis. So this is the study design. So we vaccinate HLA-A2 positive donors with yellow fever vaccine. And then for the first 2 weeks, when the cells are rapidly dividing, these people are drinking water containing deuterium. They take typically about 150 ml per day, and they drink it for the first 14 days. Okay? Then they stop drinking the water. The deuterium kind of washes away in another 2 weeks or so. Okay? And then we follow these individuals we followed them out to a year and a half. Blood samples are taken. We isolate the antigen-specific tetramer cells, okay, using MHC class 1 tetramers. And then we look at the amount of deuterium in the DNA by mass spec and by gas chromatography. Okay? And that allows us to tell how much DNA is being lost. If DNA is being lost, that means the cells are dividing. If the DNA is not lost, that means there's very minimal division. Okay? It's a very simple and elegant technique that Mark Hellerstein has come up with. So what we do is the vaccinees, all the cells that are proliferating for the first 2 weeks, all the DNA will have the heavy water, and then we follow the same cells in the same individual for up to a year. In fact, in some of them we followed out to 2 years, and then we see how much of the label is lost over time. Okay? The results actually are very, very interesting. And what we find is minimal dilution of the label. So what here on this axis is the amount of DNA that is lost over this 1 year period, and there's less than 10 to 20% loss of the label over this 1 year period. Okay? What this means is that the cells, they divide initially very fast, okay, when you get this big increase, but once the virus is cleared, okay, then basically they are very, very static. Many of them die, but the ones that survive are very static with very little turnover. And our estimates are as follows, that the label die-away is minimal with a half-life 500 days. Okay. What this means is that once you've generated a memory cell, it is sitting as a quiescent memory cell in humans with a very long intermitotic phase of about 500 days, and then every 500 days or so, there is a slow division, which most likely would be mediated by cytokines like IL-15 or IL-7. So this really is one of the first evidence of truly not only, many people have shown memory cells being there for long periods of time, but this is showing that they're not due to some continuous low-level cycling, okay, but really truly a long-lived memory cell that can be in a G1, G0 phase for up to 2 years. Okay. So let me end here by acknowledging many of the people who've done the work, and happy to take any questions.
Thank you so much, Rafi. That's impressive. I haven't seen these last.
Yeah, we were very surprised. Actually, Mark Hellerstein, who did this, was also very surprised because Mark had done a lot of these experiments, but looking at the subsets, looking at TEM cells and TEM-RA and even the TCM. Okay, they are turning over because you have no idea of the specificity, right? So unless you know what cell you're looking for, you will get a lot of background noise. Yeah. He was very surprised by how stable these are.
Great, but it's for the audience to ask. Please.
My name is Nekoe from Kenya. Thank you very much for the very detailed presentation. I'm just curious about the CD4 response.
Yes.
Because I live and work in the third world and there's a lot of HIV. So have any of those responses been replicated or seen in HIV positive individuals?
Yes, very good question. So there's a very nice paper that's published by Shane Crotty in Immunity last year. And he's looked at TFH response in HIV-infected individuals. And he finds a very nice paper which shows that HIV-infected people who have better antibody responses against HIV actually have higher levels of CD4 TFH cells. So there's a clear role for anti-HIV antibody in HIV-infected people and correlates with having good CD4 TFH responses. And then you saw, I think, a slide was shown from a very nice study that Hideki Yuno did in influenza vaccination. So again, after influenza vaccination, you see an increase in CD4 TFH cells that correlates very nicely, beautiful correlation. That slide was shown by one of the earlier speakers. Beautiful correlation with the increase in the antibody response.
Please.
Regarding, sorry, Ellie from the Netherlands, regarding the follicular helper T cells, before I understood there's also several subtypes of those.
Sorry.
Are there also subtypes of the helper T cells influenced by the cytokines, or is it just because they are in the presence of Th1 cells or Th2 cells that they behave differently, or it's really the follicular helper cell itself?
No, they're all different lineages. So
yeah,
what
so
initially drives a naive T cell to becoming either Th1, Th2 is the innate responses. Okay? The innate responses will, depending on what innate responses you have, it will either become a Th1 cell or a Th2 or TFH or Th17. Okay? And then each of those cells have their own transcription. So the innate signals induce a specific transcription program that then determines the fate of that cell.
Yeah, but before I understood, like a follicular helper cell can also have a like Th1 type helper.
Yes. I know. Yes, that's a very good question. Yes. So the question is, does a Th2 cell become a TFH cell? Okay, perhaps it might. I mean, it's an interesting, complicated question, but yes, potentially yes. Yeah. But what we have data on, also others have, is that if you have a cell which really has a Th1 commitment, which is the expressing Tbet, expressing the epigenetics of that very different, expresses very high levels of granzyme B, that cell actually is very inefficient in becoming a TFH cell. But Th2, I think there's more plasticity in the Th2 cell, yes.
So is it then a specific way, like if you design a vaccine to like maximize the, like, is it about the numbers for the TFH, or is it also the quality of the TFH?
I think both. I think numbers are critical, absolutely critical. Yeah. So the question you're asking is a very good one. Are there specific adjuvants that can give you TFH cells? And I think there's a lot of work going on in that area right now to identify adjuvants that might specifically target TFH cells. The TFH differentiation is also quite interesting. It's also heavily, and I didn't mention that, it's also heavily dependent on signals from B cells. So if the B cells are not there, you don't get the full differentiation of TFH cells. So in some ways, any immunogen that activates B cells is already halfway there in activating the TFH cells. Yeah.
There was a question in the back.
Yeah, that was also my question. Are there any adjuvants that do induce these TFH cells? Can you say that?
Well, I don't think an adjuvant so far has been defined as specifically targeting TFH cells. Okay, but of course, there are many adjuvants which give us great B cell responses, so they're doing something very nice for the TFH cells. Okay, but there's now quite an effort in several research labs in the US, and also I think in some companies now, to try and see if they could specifically identify adjuvants that would be, which would kind of put more of the CD4 T cells into the TFH pathway. One potential target there might be ICOS, because ICOS is is preferentially expressed on the TFH cells.
Okay, and the second question I have to your second part is the specificity of these tissue homing CD8
Yes.
T cells. Is that identical to the blood-borne CD8 T cells?
You mean in terms of T cell receptor usage?
Yes, T cell.
Those studies haven't been done.
Okay.
Okay, whether it's a different TCR that and usage that, and I mean repertoire that goes there versus, but I think there'll be substantial overlap, because if you start with a monoclonal, if you start with transgenic cells, they go everywhere. So it's not that only certain TCRs can go one place and not the other. But in a natural infection setting, the TCR usage of the TRM has not been studied in much detail. But it's an interesting question. Yes.
Question in front.
Yeah.
Yes.
Alba Maria from PAHO. You would say, talking about vaccine safety of yellow fever, you would say the viscerotropic disease after vaccination is related mostly with immunosuppression?
Yeah, that's a very good question. So the question here is about that even the yellow fever vaccine is one of the best vaccines we have. There have been side effects with it. And one of the main concerns has been what's referred to as viscerotropic yellow fever. That is, in a small subset of individuals, the yellow fever vaccine acts like it was a wild type yellow fever, even though it still is the wild type. It's still it's not a mutation in the vaccine. It still is the vaccine virus, but it spreads very. It's still an unresolved question, because those are usually not what you would refer to as highly immunosuppressed individuals, and most of those cases are in the elderly, but occasional cases have also been reported in the younger people. So the underlying mechanism why one in several 100,000 people get this viscerotropic thing is not well understood, but certainly is more in the elderly. So there's now a contraindication for vaccinating the elderly with the live vaccine.
I am Rashmi from India,
Yes.
and my question is from public health point of view. People who are going to endemic countries for yellow fever, and like you said that neutralizing antibody appears after 14 days. So suppose if they don't take the vaccine and just take the vaccine before leaving the country and reach there and they are exposed to the infection, so what will be their immunological response?
Yeah, I think
How
it's
will they react to that?
Yeah, I think it's a very good question. I think the recommendation always is that you take it a few weeks before you go, not the day you're going to the airport. Okay, in which case it may be a little bit too late. Yeah, so I think certainly it should be at least, ideally should be a month before, but I think even 2 weeks would be fine, because neutralizing antibody comes up very quickly.
Alexander Goldstein, Russia. Are there any immunodeficiencies identified in humans which are connected with dysfunctional TFH?
Good question. Very good question. And not that I'm aware of as a specific defect. Yeah. Again, I think a place to look might be there might be, I think there's some, actually I take back what I said. There is data with ICOS. People who have ICOS ligand deficiencies do have TFH problems, yes.
Hi, I'm Tuval. The experiment with the deuterium is really cool, and may have even implications to the decline of memory cells over time. And you showed that what you measure is the percentage of labeled cells, but that's really dominated by two factors, right? The rate of creation of new cells and the rate of death of old cells. Can you differentiate between these two?
No, what that data, so if you're losing the deuterium from there, it means the cell is dividing. Okay.
Not necessarily.
Yeah, if the cell,
you can have very little new division, but very large death in the percentage.
no, but remember, there are two different things. So this one, we're only isolating tetramer-specific cells. Okay? So the cells which are dead, we're not even looking at them.
But what you're measuring is the ratio between labeled and unlabeled cells.
No, we're not. I can explain the technique to you later. No, we're not looking at the ratio between. This is not like flow analysis. No.
Oh, okay.
No. You isolate antigen-specific cells. You know how much DNA there is, how many cells there are, and you look at the level of deuterium that was there at time 0 at 1 month, 2 months, 3 months, 6 months, and 1 year. Okay. So you're looking at the cells which are alive and seeing how much division they've had.
Oh yeah, but that's fine. But so you're looking at these specific cells, either the old ones that are labeled or the new ones that are non-labeled, right? So if you
Yes, if there are new ones which are non-labeled, then you would lose, then the ratio would be low.
Exactly, but the ratio can be low not because of high rate of division, but because of high rate of death of old cells, which you don't measure now. You only measure the live cells.
We can talk later. Let's talk later. I'll explain to you. This is something we've discussed quite extensively, okay? Yes.
Hi, thanks for the great talk. Quick question about immunosenescence in the elderly. I mean, you've got these, which T cell compartment is most affected? I mean, if you've got these slowly dying memory CD8 cells, are they relatively protected in old age, or because they don't senesce as quickly, or are there other cells that senesce quicker in the T cells?
Yes,
and how does that affect your vaccine?
So of course, in this study, we haven't done that, but work that's been done by us and by Frank Ennis actually initially with the smallpox vaccine, and Frank published a kind of a classic paper many years back where he looked at smallpox-specific CD8 T cells, memory cells in somebody who had gotten the smallpox vaccine 50 years or 60 years back. Okay? And you could still detect a small frequency of those cells. So certainly you can have, even in individuals, and then we followed up with a study, and several other people did this. So even in people who had gotten the smallpox vaccine 70, 80 years earlier, you still could find some memory CD8 T cells there. So certainly some cells can be there for a very long time, even in age of senescence. Christina, yes.
A quick question. You mentioned that the correlate of protection for the yellow fever vaccine is the neutralizing antibodies.
Yes.
But then you went on to say that there's a high level of CD8 T cells are generated and they're also long-lasting. Is it possible that T cells are also correlate of protection for yellow fever, but we never really looked at it?
Yeah, I didn't say that they don't contribute to it, but what I'm saying is that the vaccine correlate is the protective, neutralizing antibody. Yes.
And I'm thinking about that because we could.
Yeah, I'm not saying that the CD8 T cells are not important. In fact, my bias is that yes, in giving you full protection, yes, I think they would also be of value. Yeah. Yes, I know it's relevant. I know what you're thinking of in terms of the dengue vaccine with the yellow fever backbone and not having her. Yeah. Yes, exactly. Yeah, the many. Yes.
There was a question in the front.
Suzanne Hane from the Netherlands. So there are these suggestions that live vaccines have non-specific beneficial effects to counter infections. Do you know which part of the immune system is...
What do you mean by non-specific effects?
Yeah, so that's the observations in Africa that the mortality is lower in children who've had MMR as the last vaccine. And there's a recent paper from Denmark in that there's less hospitalizations in children who've had a live vaccine as the last vaccine in their schedule. And
okay, all right.
On Friday, we'll talk a little bit about that.
All right, okay. I'm not an expert in that, but I can tell you something which is rather disappointing. I think will be disappointing to people who have that data is, in all these vaccine studies we have done with the yellow fever, and also we've done quite a bit with vaccinia in humans, which is both acute viral infection and live vaccine, we see very minimal to no bystander effects. So we have very carefully looked at antibody responses to like 10 other childhood vaccines. It doesn't change at all. Only the specific vaccine goes up. That is, yellow fever vaccine goes up, response goes up, vaccinia response goes up. We've looked in them in 10 other vaccines they've got. We looked at measles, mumps, rubella, tetanus, polio. It just flat lines during that window. We've also looked at limited studies on CD8 responses to other pathogens. Okay, nothing happens there. It's only the specific response. So if there is protection to other things, it probably is by some innate mechanism, and because that's something we certainly have not looked at. Yes.
Yeah, in the slide you say about the yellow fever virus can induce CD4 polyfunctional CD4 T cells. Is this we aim for this, right? But for factors that help it to be polyfunctional? What kind determine this to make it polyfunctional than monofunctional?
Well, usually you're talking about CD8 cells. I showed the CD8 T cell data. You also get very functional CD4s, but I didn't show you that. For the goal, a good CD8 T cell will do multiple things. It will be able to become a very effective killer upon restimulation. It should have proliferative potential. It should be able to secrete interferon gamma, TNF alpha, RANTES, MIP1 beta, so on, multiple cytokines almost within minutes or of encounter of antigen. And that's what these yellow fever specific cells do. And most good vaccines and most acute viral infections will give you, it's not a unique property of just yellow fever vaccine. Most acute viral infections will give you that. Where you have problems with functionality is if you have a prolonged chronic infection. So this T cell dysfunction or monoclonal properties of CD8 T cells have been seen, especially with HIV infection or hepatitis B infection or hepatitis C, where you have a very high level of antigen that's continuously stimulating the T cell receptor. That results in dysfunction of the T cells. It's a topic that we and others have worked on very extensively.